Hi, it's Mr. Anderson, and in this podcast, I'm going to talk about these guys, bacteria. Here's a couple of E. coli that are undergoing binary fission. Bacteria is an ancient domain. They're quite a bit different than us. We're in the domain Eukarya, but they're just as fascinating. They've been around billions of years. Some of them are evil to us, and some of them are beneficial to us. And so if we look at their phylogeny, basically, if this is that last universal common ancestor, or LUCA, basically, they diverged from us a long time ago, and we have all these different types of bacteria. Know this, though, that there was a horizontal gene transfer as well. So mitochondria, chloroplast inside our cells used to be bacteria of their own, and so they went like this and became part of our cells. And so there's a ton of this horizontal transfer, but basically, bacteria is going to be this other group, and they're going to be diverse. And they fill all of these different niches on our planet. We're more related to archaea bacteria, and I'll talk more about that in the next podcast. Let's talk about the structure of them. First thing would be the size. Imagine this right here. This bacteria on your computer screen is about this big. Then eukaryotic cells are going to be about the size of you. So we're like 10 times bigger than a prokaryotic cell or a bacterial cell. A few other things that are different. Well, our DNA is going to be organized in chromosomes, which are linear stretches, and they're going to be inside a nucleus. And so inside a bacteria, they have what's called a nucleoid region. So all of their genetic information is going to be in here in this wadded up center location inside the bacteria. But they'll also have extra bits of DNA, and those are going to be found in these structures called plasmids. Their genetic material, instead of being in a line, is actually going to be in a loop. And so they're going to have a loop of DNA. Another thing they don't have are going to be introns. In other words, every little bit of this DNA is going to code for genes or is going to have a function. And then they're going to have these plasmids, which are little extra bits of DNA. But the whole thing is wound up so it fits inside the bacteria. Another thing that they're going to have is let me kind of jump ahead. As we move out is going to be the cell wall. And so if we go here, all cells, of course, are going to have cytoplasm or cytosol, and they're going to have ribosomes. However, bacteria ribosomes look a little different than ours. But if we move our way out, they're going to have a plasma membrane, so they're going to have a lipid bilayer. But outside of that, they're going to have a cell wall. And so bacteria are going to have a cell wall, and that cell wall is going to be made up of a chemical called peptidoglycan. Peptidoglycan is a polysaccharide, but it's going to have all of these cross-links between it, and so it's a really stable structure. It's going to be different than the cell wall that we'd find in plants and different from the cell wall that we'd find in fungi. And remember, we don't have a cell wall. Now, luckily, we can target this. So a lot of antibiotics, like penicillin, for example, is going to punch holes in that peptidoglycan in the cell wall. It's going to lyse the cell. And we can take a whole bunch of penicillin as long as we're not allergic to it, and it's not going to impact us because we don't have that cell wall. Okay, so they got a cell wall, and then as we move our way out, the next thing is going to be a capsule. A capsule is going to be like polysaccharide, so it's going to be this jelly-like material. And then lots of times you'll have pili that'll move outside of that, and so these little appendages on it. Now, we